All right, guys. So we are back again to continue talking about biological mutations. And now we're going to talk about mutations at the gene level instead of at the chromosome level. So we're still looking at information on how to analyze genetic information in cells and how it's expressed. That's SB2. And we're looking at B, which is that inheritable genetic variations may result from things like new genetic combinations um, through meiosis, things like non-lethal errors occurring during replication. That's the kind of stuff we're going to talk about today, and or heritable mutations caused by environmental factors. So we should be able to distinguish between different types of gene mutations, explain how genetic errors occur, and explain how errors in the genetic code can result in possible changes to proteins. So remember the central dogma. We mentioned this in our previous video. We talked about it but when we were doing transcription and translation. But the central dogma just is the idea that genes are in our DNA and that our DNA provides the information and the instruction to build our proteins. And the information always goes from DNA to mRNA to proteins. And it does that through the processes of transcription, where we make an mRNA molecule, and translation, where we go to a completely different language. We're no longer in the nucleic acids language. Now we're looking at amino acids, and we have amino acids that build a protein. So gene mutations are mutations that change the structure of the DNA during replication. They can be accidental changes to chromosomes or DNA. This can happen just by mistake. Sometimes the wrong nucleotide gets called in. Typically, enzymes like polymerase will repair that mismatched DNA sequence. However, sometimes the mutations can be missed. Mutations can be what we call silent, meaning they don't have any sort of an effect. They don't change anything. Sometimes they can be beneficial. They can be a good thing. And sometimes they can be harmful and they can hurt us. Ultimately, the effect of a gene mutation really depends on when and where they occur and if they're fixed. Most mutations involve misplacing a nucleotide in the gene segment. So instead of T pairing with A, we might misplace that and accidentally put a G in there. So sometimes the wrong nucleotide can be placed in the sequence. If that is not fixed, then when the gene goes through transcription and translation, the mutation may possibly change the outcome, the ending of protein that we get. And a genetic disorder is just an illness that's caused by one of these abnormalities, either ones that are happening in the genes at the DNA level or ones that happen at the chromosomal level, like trisomy 21 that we talked about in the last video.
The entire organism. For instance, the nucleotides GAA tell a cell to add glutamic acid to the protein it's building. If that code changes by a single letter to become GUA, the cell will instead add valine, a completely different amino acid. This type of substitution is called a missense mutation, and it creates a different protein that might not work as intended. This one-letter mutation causes sickle cell anemia, a disease affecting red blood cells. Think of it this way. Your DNA is an instruction manual that has to be followed to the letter, literally. So changing even one letter can alter the instructions. Imagine if the instructions said, make a hatchet. But because of a mutation, the H in hatchet became an R. Instead, you'd get a ratchet, and no matter how hard you try, a ratchet can't split logs as handily as a hatchet. Alternatively, GAA could become UAA, a stop codon. This code tells a cell that whatever protein it's building is finished, even when it's not. And this can stop production, resulting in a fragment rather than a complete protein. This type of mutation is called a nonsense mutation, and it's been linked to some serious diseases, including cystic fibrosis, a genetic condition that affects the lungs and other organs. There are also silent mutations, which might be a misleading name. See, for decades, scientists thought a lot of substitutions didn't do anything because genetic codes have a certain amount of redundancy. For example, GAA codes for glutamic acid, but so does GAG. So if this last letter mutated from an A to a G, nothing would happen. Or at least, that's what we thought. Until in 2022, a study on yeast found that these mutations still hurt the yeast's ability to reproduce in more than 75% of cases. Even if silent mutations weren't changing the molecules in a protein, they were affecting when and how often proteins were made. More research needs to be done to see how this applies to other organisms, but there's a chance that these silent mutations aren't so silent after all. Another way mutations can happen is if nucleotides get deleted or extra ones get inserted. These are called frame shift mutations. When this happens, the entire protein building process is thrown off. To understand why, let's play a quick imagined game. Welcome to Threeopoly, where the only rule is after every three letters, you have to add a space, whether the word makes sense or not. So, round one, translate this string of letters. I'll wait. All right, time's up. If you said the big old cat sat, you are correct. 10 points to all the old cat lovers. Ready for round two? This time we have one extra letter. And go. I'll keep waiting. That's time. So the new correct answer is though ebegold to sut. That makes no sense. This is what happens in a frame shift mutation. Since the letters in DNA are read in groups of three, if you add even one extra letter or take one away, that rhythm is disrupted, and you could end up with a completely different protein. Now, it's important to remember that mutations aren't harmful by default. They're just changes to the genetic code, and their outcomes fall on a spectrum from positive, like the one that helps you feel rested without a full 8 hours of sleep, to negative, like cancer. For instance, if you're an adult who can chug a huge glass of milk without getting a stomachache, you probably have mutations to thank for your lactose tolerance. Other mutations can protect people against HIV, prevent severe cases of malaria, and reduce the risk of heart attacks. And of course, mutations can be neutral too. They might cause a change that doesn't affect an organism's ability to survive, or even a change so small that it's imperceptible. While we can take
All right, guys. So he mentioned several types of mutations: substitution, deletion, and insertion. So a substitution is just where one letter gets changed at one place. Deletion, we lose one or more letters. Insertion, we add one or more letters. And both deletion and insertion cause that frame shift mutation that he talked about. So all mutations that are at the gene level occur during replication, can occur during replication. And they can disrupt the structure or function of the protein, but they don't always, just like what he was saying. A point mutation or a substitution mutation is just where one single nucleotide gets changed. So we still have three nucleotides to make up a codon, CTG, and another three to make up a codon. It's just GGG instead of GAG. An insertion or a deletion, we actually either remove letters or we add letters, which results in that frame shift mutation. So here, if we delete the T in the fat cat sat, and then we still read it in groups of three letters, we no longer have anything that makes sense. That's what the frame shift mutation means. It means everything from the point of deletion or insertion has changed, and the codons can be read wrong. So it's going to code for the wrong mutate, or not mutations, I'm sorry. It's going to code for the wrong protein at that point. All right, guys, that is everything that I have for you for mutations. I hope you have a fabulous day, and I'll see you in class. Bye, guys.